already delivered so many lectures in repeated systems and various and suite and other functions. He has already awarded with so many prestigious awards as well as invited speakers. So I welcome sir on this ISFCP dialogue. Please sir start your speech.
All right, thank you very much, sir. And I really appreciate Dr. Singh for this invitation. I appreciate the leadership of the college for this invitation. Within few minutes, we're talking about hard awareness, roles of a researcher. And I promise not to take much of our time because I believe that there will be questions after this presentation. And this is the outline for this presentation. We're talking about the history of use of medicinal plants, reason for use of medicinal plants, the WHO statement, that is their stand about the use of medicinal plants. We'll talk about drug discovery, the approaches, the roles of a researcher in drug discovery, and we'll talk briefly about application of all these approaches in drug discovery. Uh, One thing that I want us to know is that the use of medicinal plants has been in existence for several years, and evidences gathered over time justify this. As we could see, that the Nenanda man, the sepulchre of the man, dated 60,000 years, validated the use of medicinal plants among the elements, and 4,000 years old document, which we call Sumerian clay, was found recently. And this Sumerian clay has details of medicinal plants that have been used that time for one ailment or the other. And the use of medicinal plants is also documented by Asian, Chinese, and also in India. We have Ayurveda records as well, which justify the use of medicinal plants. So therefore, the utilization of botanicals for treating numerous ailments are greatly contributed to the development of folkloric prescription for research purposes. And we are blessed, more especially Africa and Asia, with medicinal plants, several medicinal plants. And out of these medicinal plants, only 6% have been investigated for their biological effects, their therapeutic effects, while 15%, only 15% have been uh, formulated bioactive compounds for the treatment of one ailment or the other. This justifies that we still have a long way to go in validating the physiological effects of all these medicinal plants and their constituents in order to be able to utilize, to maximize the, the, these medicinal plants. And due to this, we researchers need not to relax because uh, these plants, they are at our disposal, they are accessible, and the fact that we have them, we need to be motivated more than any other researchers from other continents. So we need to focus on these medicinal plants and think on how we can harness their potential. So now, among several reasons for the use of medicinal plants, one of the reasons is their affordability. They are affordable to us in Africa and Asia. They are accessible in Africa and Asia. And also, we have few reported side effects. Although most time we used to hear about several things about medicinal plants, that maybe the complication, this, and that. But all these are actually due to maybe poisoning. Recently, I know we've read about what happened in India about a woman that used supplements and later come down with lead poisoning. So poisoning is there, overdose is there. So I see this as I don't see overdose and lead poisoning as the as side effects of medicinal plants. I see them as maybe complications coming up from the overdose or from uh, adulteration. So plants are friendly. They are within our reach, and we have the records of their use over time. What we just need to do is to standardize and also evaluate the toxicity of this plant. So traditional medicine continues to play an important role in the healthcare system of many people around the world, including Indians, Chinese, African, American, as I mentioned earlier, and this has led to establishment of complementary medicine centers, that is alternative medicine centers, even in US and other countries, because we've discovered that we cannot run away from this medicinal plant. And even from the history, all these drugs, potent drugs that we have presently originated from medicinal plants. We'll talk about all the approaches and processes later. So now even WHO has come boldly to declare that there's need to document the use of medicinal plants in different parts of the world. And let me quickly share the results during COVID-19. If you're familiar with what happened across the world, we discovered that a lot of medicinal plants were discovered that time. And many people used these medicinal plants to boost their immunity against this virus. And to some extent, I can say it worked. So this to tell us that the use of all these medicinal plants needs to be documented. We need to move close to people that have been in this field for several years, that have been in this field for a long time, that are old, in order to get this knowledge and get it documented. And we'll talk about this later. So now, what is drug discovery? When we're talking about drug discovery, drugs will be from different sources. And like I highlighted some of it there, it could come from microorganisms, come from even human beings. There are some medicines that have been isolated from human system, maybe as as one of the end products of a particular metabolism process. And also, it could come from the food that we eat. We have nutraceuticals, and it could come from uh, what we call drug repositioning. That is, for example, a plant might have been known for as medication for treating headache. It can be repositioned later, maybe through discovery, researches, and repositioned later, maybe to treat nervous system, nervous breakdown, or to treat any other ailments later. That is drug repositioning. And also, it could come from chemistry as well, whereby a compound that is active but have low bioavailability can be worked from maybe by introduction of uh, some functional groups, specific functional groups to increase its bioavailability. That also can be done. So when we're talking about drug discovery, drugs can come from any form. But today we'll be looking at medicinal plants, phytoconstituents, botanicals. We'll look at this briefly in the next few minutes. Now, what are the roles of researchers in drug discovery? As I mentioned earlier, we have all these plants within our reach. They are accessible. Then we need to work more if we truly want to maximize their use. Now, when we're talking about the roles, one of the roles of a researcher in drug discovery is to be able to select a plant to work on. And how do we do this? We do this through ethnobotanical survey. We do survey, interact with people in the field, or people that have been using this particular way, that have been using medicinal plants in the community, try to document what they use, their comments about it, what are the things that they notice, the side effects, what the efficacy and the rest. We try to document that. And after that, identifying the plants that we want to work on is crucial because it's a serious of it's a grievous mistake if we claim that we're working on a plant and the plant is another plant entirely. So identification and authentication is very crucial. And after that, we go into extraction and isolation. A researcher in the field of drug discovery, especially in the field of medicinal uh, plants, or let me say in pharmacognosy, should be able to know, to be a master in using all these approaches, that is extraction and isolation. And also, a researcher in this field must be able to carry out physical chemical characterization and structural elucidation, which is uh, which is crucial in drug discovery. And also biological screening, all those biological assays, we must be able to master all these biological assays and be able to find the one that we can use to justify the folkloric use, any folkloric use of the medicinal plant. And also molecular modeling to develop new analogs too. We do this on system. I believe you've read about molecular docking before, trying to see, to measure the strength of a compound in binding with a ligand. So that one too could give us a clue about the bioavailability of the drug, and also how we can improve its efficacy. And after getting all these clues uh, in silico, then this information can be used in derivatizing this drug in a lab, and that's what we call synthesis. So all these things, all these roles, they are important. At least a researcher must have an idea of this whenever he or she wants to design uh, a study. Now let's look at the interconnectivity between all these roles that we're talking about, the approaches that we're talking about. We could see we have primary assays, and all these primary assays, at times we call them preliminary assays. They have high throughput, and they are economical, and they are not most times they are not time consuming. We call them preliminary assays because they are the assays that we carry out first before embarking on the main assays. One of them is any antioxidant study, any in vitro antioxidant study that you might think of, or qualitative or quantitative phytochemistry, which is through TPC, TFC, SOD, all those assays, they are primary assays. Then we have secondary assays. But I must say this, even we have some primary assays that can give us information or clue or directive about the efficacy of a plant. Of a plant. That is, there are some assays that we can use. There are preliminary assays that we can use that we act as the main assay for the study. And also we have secondary assays, whereby we'll be talking about all these biological assays that we do carry out in the lab, the ex vivo study, the enzyme related study, and the rest. So those are the secondary assays that we have, and this can give us lead. That is information about the lead compound, about bioactive compound that are bioactive. And then when we have a compound or a medicinal plant that is active, the next thing is to find out or to investigate the constituent that is actually exerting that therapeutic effect, and that will lead to isolation. We have all those uh, approaches that we do use in pharmacognosy, and also structuralization is key because we'll be able to get the structure of the compound that is active. Then this compound will lead to designing of interesting biomolecules, with either to increase the bioavailability or to reduce the toxicity of this plant or of the constituent. And it involves derivatization. You derivatize, then after that, we can go to clinical. That molecule can be subjected to clinical trials. And one thing is that I'm showing us this chart for us to know that clinical trial is very long. It's a very long process. We have phases, three different phases, and all those things. But what I want us to know is that there's something we call the reverse pharmacology. Reverse pharmacology is what we use in herbal medicine to actually. The advantage of reverse pharmacology is that it shortens the process of clinical trial. And what it simply means is to develop pharmaceuticals, to develop drugs from existing traditional medicine. And the fact that people have been using these drugs, have been using the medicine, the medicinal plant, which is actually what we refer to as raw material in drug discovery, in drug herbal medicine drug discovery, is because the fact that people have been using it over time and validates its uh, non-toxicity to some extent. And that process shortening the reverse pharmacology, shortening the process of clinical trial. And that is why in the market, it is, let me say, in medicine, it is easy to actually patent uh, herbal medicine. It's easy because in most countries, the agency that is in charge of this, the first thing they check is just to check the toxicity and then later on the efficacy. So, and the fact that people have been using, we've been using this medicinal plant locally justifies toxicity to some extent and it makes the process to be shorter. Now, let's talk about application of these approaches. Here, I will be fast because I discovered that our time is fast spent. So now, uh, I said whenever we want to start a study, we start with a survey. This is one of the survey carried out some years ago to document the medicinal plants that a particular community in Nigeria used to treat female infertility and menstrual disorder. And from this, we got a list of close to 30 medicinal plants with different parts in use. I just highlighted some of them. One of them is Astonia bonei, Bacella alba, which is a vegetable, Glyphar brevis, and also Kigelia africana fruit. I highlighted at the end of this presentation, who would know why I highlighted these medicinal plants out of the 36 medicinal plants documented. And also, another approach that we can use, which are found to be effective, is to relate with uh, like I mentioned earlier, people that have been using this plant, traditional medicine practitioners, we need to relate with them. This is one of our study we carry out here. We related with just one of them. We uh, we collected information about medicinal plants used for the treatment of low sperm counts in that community. And these plants were selected from we call it records there. They have their own way of keeping records, of passing it from one generation to another in written form. So we decided to go through this record and we discovered 13 medicinal plants that could be used to treat low sperm count. And unfortunately, out of these 13, only six have been investigated in the lab. That is, there's no any scientific record or scientific validation for the use of the other medicinal plants. So this we can use in order to document this knowledge so that we can pass this knowledge from one generation to another. And we have some other parameters that we can use or that we can consider whenever we want to carry out botanical survey. They are descriptive statistics, the use value index, fidelity level, knowledge index value, use knowledge index, ethno-medicinal income index, tissue importance value, ethno ethnobotanical richness, and also name homogeneity index. these are some of the material of the criteria that we can use if we want to carry out a sound and a robust ethnobotanical survey. Now, one of the plants, which is glacial brushwoods, we try as much as possible to generate uh, scientific parameters so that it will be difficult for adulteration. We have some plants that looks alike, and in order to differentiate from these two plants, we need some parameters, although they might look alike physically by under a naked eye. But under microscope, there are some parameters that are peculiar to a particular plant. And this is what we did with glacial breakfast sleeve, in order to see all these the parameters that differentiate this plant from other medicinal plants. And also, we need to, as a researcher, we need to generate a data bank for the phytoconstituents in all these medicinal plants. And even this knowledge is now useful in chemotaxonomy, whereby the chemical constituents of a plant is being used to classify them into families. And therefore, even as a researcher, we must know or be a master of all these uh, all these methods of identifying phytoconstituents. And that's what we did with Petiferia alese here. We use GCMS to to identify the constituent of the fractions, the methanol hexane, the hexane methanol fraction, and also the bonatide oil from the plant's leaf. Another approach is that after getting these fingerprints, there are some studies that we can carry out even without going to the lab, and that is in silico study. Here we try to carry out this. Uh, we from the literature, we discover that from Trichosia subcortata, only 4 compounds have been isolated and characterized. Therefore, we try to see the potential of these 4 compounds in treating several diseases, and we use in silico method. And from this, we discover that the constituents, these 4 compounds that have been isolated, might be the one responsible for all the of, for all the therapeutic effects reported so far for this plant. And we also discover some new uh, ailments that these compounds or these plants might be useful in treating them. And one of that is these plants can act as an inhibitor of kinase and nicotinic alpha beta receptor agonist. Now, I mentioned earlier that we have some preliminary studies that can eventually provide information about the bioactivity of the plant. So here, Philanthus filicalis was subjected to GCMS and also all those preliminary assays, that is antioxidant assays, SOD, FRAP, then also the in vitro and in vivo anti-diabetic uh, study too were also carried out on the acetic fraction of these plants. And from our findings, we discovered that this plant contains a lot of terpenoids that might be effective in uh, treating or alleviating diabetic condition. And also, uh, the antioxidant and the antiproliferative effects of Kigelia africana too was also carried out some times ago, where we isolated 6 compounds from Kigelia africana. One of them, which is compound 4, happens to be a novel compound, but it does not have any derivative because it does not have any activity on any of these 2 cell lines because it's a sugar moiety, sugar derivative. But out of the compounds isolated, we discovered that 4 of them are cinnamic acid derivatives. They just have different functional groups at different positions, which also have effects or which also have exact different effects in their efficacy. Likewise, we try to see the effect of gallic acid in alleviating cadmium chloride toxicity. And we discovered that gallic acid could actually protect the nervous system, that is, the nervous system from the attack of oxidative stress. And even as well, we try as much as possible to evaluate the anti-diabetic effect of glacial breakfast as well in vivo and in vitro. And just to open our eyes to why Sphilanthus filicalis tapinois could be a potential. It was validated there because we discovered that the N-hexane fraction of this plant inhibited most the alpha amylase activity. It inhibited the, the activity of alpha amylase enzyme. And from the literature, we later discovered that terpenes and terpenoids, they do inhibit, they've been discovered to inhibit alpha amylase activities. And also, last year, we tried to see the effects of Annona muricata, which we know call here as referred to as shell shop in Nigeria. The plant is sweet, and the fact that it's sweet, many people think it's not good for people that are diabetic. So we decided to investigate this to know the effects of these plants. And we discovered that even the plants upregulate P13AKT gene regulation, which is even beneficial to people that are diabetic. And also, we try to see the effects of Tetracerra potatoria on people with polycystic ovarian syndrome. And since it has been mentioned earlier in one of our survey as medicinal plants used in treating reproductive female infertility and even menstrual irregulation among women. So we decided to investigate the effects of this plant on PCOS 15, and we use albino rash. We try to design a model which is to induce PCOS with uh, letrozole. And from the plants, we also isolated for the first time, although the compound is not novel, but we isolated it from these plants for the first time, and the compound is apigenin. So also we subjected the fractions and this compound to entity assay using CHO cell line and HeLa cell line. And the reason why we use this is because it's been reported that people with victim of PCOS are 2.3 fold the risk of them having cancer is very high. That is, they are more predisposed to cancer than women without PCOS. So we decided to do this, and we discovered that these plants could be a candidate, and even the apigenin could be a candidate in developing a drug or a medication for the management of polycystic ovarian syndrome. And we also subjected Bacella alba, which happens to be a vegetable. And one interesting thing about this vegetable is that in some parts of Nigeria, whenever a woman is preparing for a lady is preparing for a marriage, they cook uh, this Bacella alba. They prepare the Bacella alba soup. Bacella alba is a vegetable. They prepare it often and often and give it to this woman in order to prepare her for childbearing. So we decided to investigate this, and we discovered that these plants too alleviate polycystic ovarian syndrome in albino rats. And also from Kigelia africana, we carried out a study, and we started with isolation, and we were able to isolate a novel hydroxyflavone derivative, a novel one that has not been reported before. So we isolated it and we subjected it to in vivo and in silico anti-cancer study. And we discovered that this compound have potential on CHO cell line. And in order to know to understand the process, we subjected it to in silico study using P53 as a ligand. And we discovered that this compound binds to this ligand, which justify that this plant could also be used in preventing or in protecting PCOS women against cancer. You know, I've mentioned earlier that they are more predisposed to having cancer. And also, cinnamic, you know, I told you that we isolated cinnamic acid derivatives from Kigelia africana, and therefore we took some of these compounds to the lab and to derivatize. And from derivatization of just one compound, we got nine derivatives, and two of them happen to be novel. So we took one of the derivatives to actually see its effect in alleviating oxidative stress in vivo, ex vivo, and in silico approaches. And we discovered that these particular compounds alleviate, protect against oxidative stress. Uh, and also, you know, after carrying out all these studies, what we just need to do is to gather all this information and make it available. For example, we can review all our studies and present it as a review paper, which will make it it has its own advantage because a lot of information about what we've worked on will be available. And anybody that wants to get the details and the procedure and everything will have to go and check our publication. So this is one of our review. I will try to review all plans, our study and other people's studies too, to actually document the effects, different effects of medicinal plants of female reproductive system. And we reported close to 7 effects of these medicinal plants. And also, you know, we tried to look at the Carica papaya. I presented these two reviews for us to understand that even at our leisure at home, when we are not in the lab or when we are in our office, we can come up with ideas and they are publishable as well. So we came up with the idea of documenting antidiabetic potential of Carica papaya because from folkloric uses to product development. And it was published. Now, I want to conclude with this. We have several approaches that we can use as a pharmacognosis. One of them is in silico study, ex vivo study, in vitro study, and in vivo study. And also we can write a review as well. But the four approaches, we need to have understanding of it so that we'll be able to apply it whenever the knowledge is needed. So there's urgent need for more translational research to determine the effects of bioactive compounds from medicinal plants in clinical settings. And also optimal concentration at which medicinal plants and their compounds are both safe and effective should be investigated and documented. And also other techniques should be applied as well. Like I mentioned, the in silico, the in vitro, and ex vivo study. All these techniques should be applied in order to validate the folkloric use of these medicinal plants. So thank you very much. I appreciate you for your patience. Thank you.
Yes, sir. Sir, first of all, thank
Okay,
you
so
so much
Dr.
for
Singh, over to you, sir. I don't know if we have any questions.
yes, sir. Sir, uh, Here are some queries from students. Just wait and watch. Sir, I want to take a screenshot. Yes, sir. Sir, question
Sir, I
is,
don't know if we have any question. I don't know
yes,
if
sir.
we have
Sir,
any question.
sir, students are asking that how the herbal concept can be future for the medicine.
Hello, sir. I can't hear you very well.
Yeah, sir, question is how the herbal can be future for the uh, medicines.
The method, sir.
Yes, sir. Sir, question is how the herbal medicines can be future for pharmacy or medical field.
Okay. One thing is that in pharmacy we have different scopes and different areas. We have pharmaceutical chemistry. We have uh, pharmaceutical microbiology, and we also have pharmacognosy. So one thing is that the foundation of pharmacy is actually pharmacognosy, if I can say, because uh, medicinal plants are the major sources of bioactive constituents that we have presently in the world. Although we have other sources, like I mentioned earlier, but medicinal plants are actually the major sources. And considering the fact that 70% of the entire world population depends on medicinal plants now, is an advantage. So we as a pharmacist should be able to dive into this, maximize this, and come up with uh, a lead that can be formulated or that can be later developed into drug or into medication. So pharmacists have a lot of role to play in this. And I need to stress this, we have a lot of role to play in this because we have everything in our hands, and the fundamental knowledge that we have, we need to start applying them now. More especially pharmacists in Asia and in Africa, we we have advantage over others, which is accessibility of all these medicinal plants. So we don't need to relent, or we don't need to find. We've been taught several. We've been taught several orthodox medicine, their applications, their use, all this and that. But we should dive into our own traditional knowledge of medicinal plants. And I must say, one of the leading countries in this field is India. Because anytime you hear Ayurvedic this, Ayurvedic that, this one, this, that. So we have the information. All we just need to do is to document this information, the folkloric uses, then validate each plant. Validate the
Yes, sir.
folkloric uses, each folkloric use mentioned for a particular plant. And from that, sky will be our limit. So we have a lot of room to
Yes, sir.
play. Thank you, sir.
So thank you so much, sir, for your wonderful talk. Sir, in very precise manner, you have discussed various as well as
Aman sir, your voice is not audible.
yes, sir. Audible now?
Yes, sir, now audible.
Yes, sir. Sir, I would like thanks to sir for their
Yes, it's
wonderful
audible
talk.
now.
Yeah, yeah. So, sir, once again, thanks a lot for the session or talk. Sir, audience have enjoyed your topics, which will be helpful for their clearing their concepts with the help of pharma as well as herbs. So, thanks a lot, sir.
I'm very grateful, Dr. Singh. I'm very, very grateful for this. And I want to appreciate everybody. It's not